Good afternoon, everybody. Thank you for coming. I hope you enjoyed your lunch. So it's a great pleasure and an honor for me to introduce our first plenary speaker for this afternoon, Professor Luisa De Cola from the University of Milan. So as you very likely know, Professor De Cola is a very prolific scientist and researcher. She has won many prizes. I mean, there's a whole bunch here, but I just want to introduce some of the I guess, perhaps more important ones. She was nominated Chevalier de la Légion d'honneur by the President of the French Republic. She's a member of the German National Academy of Sciences, Leopoldina, Accademia dei Lincei, Fellow of the American Institute for Medical and Biological Engineering. And most recently, this is very fresh news, Professor De Cola won the Ziegler-Natta Prize from the German Chemical Society. Yeah, I think I said that right. Anyway, it is without further ado that I'd like to present Professor De Cola. Thank you very much for joining us at SHIFT.
Thank you, Fiorenzo, for the kind introduction, and good afternoon, everyone. It's really a great pleasure and a privilege to be here at the SHIFT. So I heard about this conference several times, and I am really honored to be part of this community today and in the past and follow days. So what I would like to share with you today is a story that indeed started some years ago, and has to do, of course, with light and small molecule. In particular, I would like to show you how we are interested in how the molecule can essentially self-assemble in specific structures, and what are the outcomes of these self-assembly processes. And so in this case, we will see essentially that light can be used to visualize a process like the self-assembly, and of course, light will be used to detect the type of structures that will rise from this self-assembly. But during this process, we had a small problem, as many of you noticed and was mentioned already several times in this conference, that often we go through transition states that are essentially not possible to isolate. So these are, of course, transient species or out of equilibrium species. And we were wondering, how can we block this process and eventually isolate this transition species and this out of equilibrium species? And I will show you that we did in a very special way, using some kind of cages, like you would imagine, to capture essentially this system and bring them out of the solution and eventually use them for other things. And doing that, we realized also that the self-assembly could be done not only in solvents, like we normally do in water or in organic solvent, but can be also be monitored in real living systems. And so I will show you the self-assembly in vivo, not because of the imaging in this case, but because the self-assembly causes a series of biological events that leads eventually to the regeneration of the animal. So it's a very special thing, and I think you might find it interesting. And since I like very much the title of this conference, SHIFT, that means many things, as you can imagine. And for me, SHIFT in this case means today a shift from basic research to some kind of applied research. So I want to show you for the first time some results that just been patented and just published in JACS. Related to the use of light for detecting pathogens. And this is a spin-off that we created a couple of months ago. So let me start with this small molecules story. And this small molecule for us are platinum compounds, in particular platinum-2 complexes. And why did we use platinum-2? Because they are square planar molecules. And as you might know, when we coordinate ligands, like conjugated ligand to this small platinum, we can have a phosphorescence coming out from the compound. So we call normally luminescence in general terms. And this luminescence, as you can see here, can be really tuned, even just essentially flipping the ligand around the platinum compound. So this is quite interesting, but the most interesting thing comes indeed from the fact that the platinum-2 compound are square planar, so they are flat molecules. So you can imagine that at a certain point, perhaps these can start to interact and forms indeed some kind of aggregates. And if this is happening, what happens is indeed that you can create even a bond between the platinum units, so the metal-metal bonds or metallophilic interactions, that leads to the stabilization of these aggregates up to a point that you can isolate these micrometer or even millimeter structures. But not only that, because in the moment you imagine that these molecules approach to each other, something more subtle will come, because of course the orbitals that are involved in this interaction will create a new excited state. And just one pill of photophysics to tell you what's happening in this case, because as you can imagine, if we have just the platinum compounds in a solvent, and this will be essentially solvated in this situation, so they are monomeric species, if I shine light on these compounds, you immediately recognize that we can have normal pi-pi star transition or metal-to-ligand charge transfer transition, something we studied indeed already in schools. So this is quite common for this type of system when we have this conjugated ligand. But like I mentioned to you, if we have this conjugated ligand, maybe this pi-pi stacking can occur to a point where the d zeta square orbital, that are the orbitals perpendicular to the plane of the platinum compounds, will start to interact. So if you imagine two molecules now approaching, you understand that this orbital will interact with the other molecule, and so this will destabilize the d zeta square. And so what happens in this case is that the energy, of course, will rise, and now you notice that we have new possibility in order to essentially absorb the light, and a new state called metal-metal ligand charge transfer can be created. Why I'm telling you that? Because as you see already from very simplified diagram, the molecule might change color, and this is reflected not only in the absorption but also in the emission properties, and change dramatically the properties, because you can imagine that in the moment I pack the molecule will be much less sensitive to oxygen, so they will not be quenched by oxygen, even though the lowest excited state is a triplet, long-lived excited state. And then we change the nature of this excited state, going from pi-pi star to metal-metal ligand charge transfer. So with this in mind, indeed, a long time ago, we started this research with the idea to make compounds that would favor this type of self-assembly. So we made essentially compounds that were essentially neutral, using these pyridine tetrazole ligands. And what we noticed indeed, that upon aggregation, we could reach quantum yield of emission close to unity. So with this idea in mind, that was essentially very, I would say, counterintuitive at that time, we made organic light-emitting diodes based on this phenomena, that is the aggregation-induced emission. But later on, we realized that this aggregation could be also triggered by the choice of the ligand, because you can imagine that now if we switch from tetrazole to triazole, we can also functionalize this ligand with different groups and making this ligand even more hydrophobic than before. And then attaching to this pyridine that is essentially needed to complete the coordination sphere, we can attach hydrophilic tail. So essentially, we are creating a surfactant. And all of you know that a surfactant can lead indeed to self-assembly system in solution. And this is indeed the case also for this molecule that is very soluble in organic solvent, so for instance in dioxane. But in the moment you add water, of course, the system will start to aggregate, because these hydrophobic units will tend to stack. And as a consequence, you see this huge shift in the emission properties of the compound. So you go from a blue emission to a deep red emission with very high quantum yield. But doing this, during this study, we noticed something quite interesting, that these quantum yields were a little bit tricky to study, because essentially the number of photons we were giving were not essentially taking out only as a blue and red emission. There was something that we could not see in between, that was essentially a transient state that by steady state measurements we could not detect. And so Alessandro Liprandi, that at that time was a PhD student in my lab, decided to look at this phenomena using a confocal microscope. So he did essentially time-resolved experiment to really see if we could catch the event of the self-assembly process in real time. And this is indeed the first movie that he made, where you see that indeed in this condition of water dioxane, you see first these red dots appearing, and then these green rods that grow, until, of course, the system evolves, like any system, towards equilibrium, towards a thermodynamic stable state that are these blue emissive fiber. So this is quite exciting, because what you are watching with me is essentially a real-time self-assembly of small molecules. And of course, we were lucky, because each state has a different color of emission. Of course, if you change the ratio between the dioxane and water, you will find out that the system evolves even faster. So the red dots disappear almost in no time. But also the green rods, as you can see here, they are transformed, of course, in the thermodynamic stable blue emissive fiber. So this was quite exciting for us, and we could explain what was going on in this paper, because we could measure separately the lifetime and the emission properties of each species. And of course, for two of them, that are essentially what you saw also in the steady state, we could measure the distance between the platinum units and correlate this energy of the emission with the electronic interaction within the platinum units. And so what we found out was that for these red dots, the interaction was very strong. It was, the distance was only 3.2 angstrom. But for the blue part, we measured something above 4 angstrom, so no electronic interaction between the platinum occurred. We were lucky, because we were able to publish a follow-up paper with other compounds, where essentially we also made the crystal structure indeed of this unit, where you see indeed the reason why the distance is so long, because this glycol chain kind of wrap around, forming a nitrogen bond between the amino and the oxygen of the glycol chain, and therefore preventing the platinum to get together. So this is quite exciting, because essentially we can use the light at this point to see and eventually understand this process of self-assembly. And I want to show you that, just to wake you up after lunch, that when we sent this paper indeed to Nature Chemistry, the editor asked us for the cover. And this was the time, and maybe some of you have seen this movie, that was Star Wars, where you probably know this famous battle between the bad guy and Yoda and the good guy that was Lucas. And perhaps you notice already the colors of their swords. And so with this in mind, indeed, we designed the cover that was not published. Because it was not published because the editor thought that the Lucasfilm would essentially complain about that. And so they published, unfortunately, only the original confocal picture. But that's okay. I mean, I think it's good enough. So anyway, so at this point, I want to tell you something further that came out during the discussion with my students about how can we really visualize these intermediate green species. And of course, these are species out of equilibrium. We know very well that these species can live seconds, maybe minutes, but they are gone. Sometimes they cannot even be detected with a regular spectrophlorimeter. And so we were wondering, what can we do to trap these green species and eventually to let it to go towards the equilibrium in a completely different media? And of course, if you think in a very naive way, like I normally do, I think, well, I have to catch it. So I have to have something that essentially catch from the solution these green species and maintain the capture. So it must be in a confined space. And this was indeed the idea. The idea was, can we imagine to catch these unstable species and then eventually stabilize and eventually release them in a different situation? And what we decided to use is something that we are very familiar in my lab, that are silica-based nano cages. So these are, I will show you in a moment the structure. These are exactly what you see here in this cartoon. So they are cage compound, where essentially we have openings that are large enough to allow some of this aggregate to be captured. But eventually they can break on demand and release again this intermediate state. So what did we do then? I want just to show you what are the cage look like, and this is a beautiful TM, and I have to really thank Professor Patrick Schulz for, at the beginning, really computing by simple rely on computational data with 10,000 frame, and he predicts indeed the morphology of this silica material. And the silica material is quite unique because later on we could do tomography, so we could really visualize the real morphology, because he has a single pore, and he has this opening of this 3 to 4 nanometer. So it's a confined space, but it's still accessible from the outside. So it's indeed what we call nano cages. But I have to tell you one more thing, because this is not only silica, it's what we call organosilica. That means that during the synthesis of this material, we introduce some kind of reactive groups in order to make this silica degradable. Well, degradable is not the right word. I should be more precise. We should say breakable. Because indeed, our idea was to break this blue box in order then to break in pieces this silica. And what we use is this very simple chemistry, is a disulfide bond in between indeed the silica units. And all of you recognize that if I reduce the disulfide to thiols, I can break in pieces indeed this material. And maybe most of you know that the reduction of the disulfide is a very, very simple reaction and very low energetic, very low, because the reduction potential is very low, and can be done into the cells, is what happens all the time into the cell, through a biomolecule that is glutathione. So with this in mind, we thought, okay, can we really capture this intermediate state from the solution, so we can work in a condition where this is stable at least for 1 hour or 2, and then we entrap essentially these aggregates into our cage and look how they eventually develop, or they tend to go to the equilibrium in a different media. And this is indeed just a summary of this paper, where you see in the normal case, of course, in the absence of the cage, the system evolve within already 1 hour. But when we capture the system, you see very nicely that the system is stable for more than 24 hours. So we can stabilize this out of equilibrium species for a very, very long time compared with the lifetime of the original system. At this point, we thought, well, this is fantastic, because the compound is completely water insoluble, and we have a way through the cage to bring it in water, but even more than in water, to bring it inside cells, and then look eventually at the evolution, at the polymerization of the system inside the cell. And this is indeed what we have done. So we essentially incubate our nanocage in HeLa cells, where you perhaps can appreciate these little dots that are indeed the cage with the yellow compounds. And if you wait until the cage breaks in pieces, that is about 6 hours, you see the formation now of the polymer inside the cell. And this is a very powerful concept, because, of course, can be done not only by supramolecular chemistry. You could imagine to have a very reactive intermediate that cannot be indeed internalized into the cell, or could be the beginning of a much bigger molecule. So it could be a way, essentially, to transport and release in a special way this material. So if you want to know more, this was also published in JACS, and I got the cover. And the guy who did the work is now at Technical University in Munich with Angela Cassini, Guillermo Moreno Alcantar. So at this point, we were wondering, well, this is fantastic. We can go in cells, the compound is not toxic. What about going in vivo? And will in vivo this self-assembly work? And now the question is, which animal will allow us to follow the self-assembly in vivo? And of course, we went for transparent animals. So typically, a more simple system like what is called Hydra vulgaris, that is these little polyps, where you see essentially that is fully transparent. It has this branch. The biologists like it very much. It's a a freshwater animal, and is very well known because the way it reproduces both sexual and asexual, but also is studied because he has an exceptionally regenerative capacity. So in other words, if you cut one of the branches, the animal is able to reproduce the branches. So we knew nothing about these animals, so we teamed up with a group of Claudia Tortiglione in Naples that studied this animal for other reasons. But I have to tell you one thing about these animals, that essentially is constituted by 2 sets of cells that are essentially constituting the interior part and the external part, that has the endoderm and the ectoderm. And what is important for you to know is that the regeneration of this animal is essentially triggered by systems that are nematoblast cells that are able to produce the stem cell that eventually will be differentiated into the animal in the different organs. Of course, we have to make our compound water soluble at this point because we cannot give to eat to the animal our water insoluble compound. So we design a similar system that still go to self-assembly in water, as you can see here from this beautiful orange emission. And of course, then we decided to look at the toxicity of this compound inside this animal. And what we noticed was something very nice. The animal essentially can tolerate very nicely this compound up to a concentration of 50 micromolar. And I want to show you the picture because I think they are very beautiful. So these are the hydra that are untreated. And of course, when you start to add the platinum, you see that the hydra feels well, I would say. Up to 20 micromolar, everything is normal. But at 50 micromolar, the animals start to suffer, and eventually, after 4 days, they die. So essentially, we have to stay below this concentration in order to make sure that the platinum that will be ingested by the animal will not kill the animal. What does it look like, the animal, when they eat the platinum? Well, they look very beautiful because they light up like a Christmas tree, essentially. So the emission quantum yield that has been only estimated because in water is not so high, so it's about 50%. And you see very nicely that the aggregation is everywhere. So essentially the platinum is distributed both in the branch, in the head, in the body of the animal. And we were wondering, is the platinum going everywhere, in every type of cells that I showed you before, or it prefers to accumulate in certain type of cells? And what we noticed was essentially that the platinum was present both in the endodermal cell and in the ectodermal cell, but it was highly present in these nematocytes that I mentioned to you at the beginning as responsible for the stem cell production. And of course, we can calculate the amount of platinum that is in each animal, so we can do ICP mass in order to evaluate how much of this platinum is ingested by this animal, so we can have a real understanding of the amount of platinum that gives this self-assembly. But the most fantastic results came out, indeed, when the people were studying the growth of this animal. This animal takes about 3 days to start to grow. But what we noticed was, and this was 100 times repeated, that when we give even a very small amount of platinum, the animals start to grow at day 1. And this was weird. I mean, why would an animal start to grow when we give, in principle, even a toxic compound? So at the beginning, we thought, well, it's because of oxidative stress. So the animal feels stressed by this platinum compound that is not what they would like to eat, and therefore, they accelerate their metabolism. But this is not the case, because then if you look at the lifetime of this animal, it's normal. So it's just, this acceleration that is amazing for all the animals we tested. And so at this point, we start a very long journey in the biological event that could trigger this, of course, this growth. And the first things the biologists did was indeed to study the ratio between the stem cell and the normal pluripotent stem cell. So that what they measured essentially was with the control that was here in the blue bar, how much this stem cell production increase when they give indeed the animal the platinum compound. And this was very unusual because they were essentially noticing that more than 30% more stem cells were produced into the animal due to the presence of this platinum compound. And then finally, we decided to investigate by transcriptometry what was the gene that was creating this stem cell production. And the gene that was isolated and studied is this gene that is called HYMIC-1. It's a very important gene because it's a gene that negatively regulates the stem cell self-renewal, and is also present in vertebrates, like in humans too. And this, in case of the human, is regulating the metastasis, so that the growth of tumors. So what we noticed was that while for the untreated animal that we consider as 1, the HYMIC-1 was regulated in this way, in the case when we give the platinum, we have a strong downregulation and therefore a strong production of this stem cell. At this point, the marine biologist said, well, probably we have other events that are happening in this animal because of this platinum system. And what they did was to measure indeed also the enzymatic activity of the most important enzyme of this animal. And these are all enzymes that are related, of course, to oxidative stress. So what they noticed was that both the superoxide dismutase and also the catalase and putative peroxidase were extremely overexpressed. So the enzymes were working much better, let's say, in the presence of this platinum. And this is unusual because this happens normally when you give as a I would say, integrator, a very strong reducing agent. So essentially, they were noticing that there was a redox behavior that was not normal. And so at this point, we studied in detail what happened to the platinum that is entering indeed these nematocytes. And we were very lucky because thanks to the luminescence of the platinum, we could really follow in real time the formation of this nematocyte, this charge that is responsible essentially for the stem cell production. So we could really follow the filling essentially of the nematocytes and then the excretion of this collagen type system that then is causing all this cascade reaction. But not only that, what we noticed was indeed related to the redox properties of the system, that in the untreated animal, the battery cell that you see here, that are all very nicely organized, becomes completely disorganized when we add the platinum system. Analogously to the fact that if you add a DTTT, that is a normal reducing agent, happens into the animal. So we explain this phenomena by the fact that when the platinum self-assembly indeed changed dramatically the redox properties. So whatever I show you today is essentially related not to the platinum compounds, but really to the self-assembly system, so to the supramolecular interaction in vivo of the system. So this is a general phenomena. Well, we tried on a different animal, that is the Nematostella vectensis. This is also a transparent animal that lives in seawater. And this is similar to zebrafish. Most of you know that zebrafish is formed by a single egg that then is transformed through a series of transformation and eventually form the animal, the adult animal, in about 10 days in this case. And so we did the same experiment. So we feed the animal with this platinum compound. And what we noticed, and I don't want to go into the biology, but just on the picture, that the untreated animal, of course, after 6 days is not formed, because normally it's not formed, but with the platinum, it is already formed. So again, we have this huge acceleration on the formation of the animal when we feed with the platinum. So that's where we stand. I don't have many other explanations, but it's, of course, a very intriguing phenomena of self-assembly in vivo that could lead to many other research, not only with platinum, but with other systems. So for the last few minutes, I want to give you now a shift, like we were discussing. And the shift is indeed from lab to company. And this is something we are working in my lab since many, many years. But it's the first time I present this paper, and this was mainly due to patents. And the idea is quite simple, essentially. And this also the basis of 2 European projects that follow up from the very first idea. That is, can we develop a portable device that is based on amplification of a signal, able to detect the full pathogen? So all of you know, so that because of COVID, that when we do, any type of pathogen detection, we have to extract the genetic part of the pathogen. So we have to extract the DNA or RNA, then amplify the DNA, and then finally we have, by PCR, the amount of pathogen. And our idea was, can we avoid all of this? Can we imagine just to catch the pathogen, the bacterium, the virus, and tell you in real time, in 2 hours, what is the pathogen and how much do we have? So how can we do that? Well, I have to make a kind of a long introduction. I'm sorry for that, but I hope you find it interesting. And the detection system at this point is not fluorescence. And so in this case, we will use the light not to see, but we will produce the light in order to sense what we are looking at. How do we produce light? Well, all of you know that if I shine my laser on the molecule, as I showed you before, we can produce light by emission, fluorescence or phosphorescence. Most of you know also that, because all of you have a telephone in your pocket, that we can produce light by electroluminescence, by charge recombination. But maybe not all of you know that there is another interesting way to produce light that was discovered by Alan Bard in the 70s that is called electrochemiluminescence. So in other words, instead of having two electrodes, we have only one electrode and then a reagent that does the other semi-reaction. And this is a very interesting concept because essentially we can generate light by electron transfer between species that are generated electrochemically, where one can be an electrode, the second one could be also a chemical species. Why this is so powerful versus fluorescence? We always think about fluorescence as the most sensitive detection system. In reality, it's much more sensitive than light excitation because, indeed, we don't use any light source. So essentially, we are looking at the emission, generating the light in the dark. It's like, if in this room I switch off all the light and I turn on a candle. If I turn on a candle, you will see the light. But if I turn on the candle in a room with millions of light, you will not see the candle because you are already essentially filled with the light of the room. And so having no light source means also that we don't have a very expensive device like lasers or special lamps. We have no light scattering because we don't use light excitation. We don't have any background because our background is the dark, is just the things without light. We have a large range of linearity, and we form the excited state in a controlled way because we need a potential that we must apply, and the potential is typical of our dye. So it can be very selective in a very complex matrix. It's ultra-sensitive, so it can be sub-picomolar detection, and probably all of you know that when you do a test like your blood test, most of these are done by ECL analysis. And so you can quantify proteins, hormones, enzyme, toxin, DNA. Now recently, Roche put COVID assay, and so on and so forth. So what is then the principle of this ECL? It's very simple. So Roche, that is the largest company for ECL, and many of the companies in China, works on metal compounds. In particular, they work on ruthenium compound. This is a very simple molecule, ruthenium-3 bipyridine. And then they will have one electrode, and the second electrode, I told you, is not an electrode, it's a chemical species. That is a tripropylamine. It's a very simple small molecule, quite toxic and volatile, that will be essentially our electron donor. So how does it work? Just very simply, so we have an electrode, and we have a ruthenium that is attached to an antibody. We do a sandwich assay, so this is our analyte that will be sandwiched between the antibody that is fixed on the electrode and the antibody that is connected to the ruthenium. Here, of course, electron transfer cannot occur because the ruthenium is too far from the electrode. The antibody are nanometer species. So what is mediating this electron transfer is indeed our tripropylamine, because the tripropylamine that is added in this assay can be easily oxidized at the electrode. It forms this radical cation, it can lose a proton, and this radical can indeed reduce the ruthenium-2 to ruthenium-1. At this point, this ruthenium-1 can be reoxidized by the tripropylamine radical cation to the ruthenium excited state. And in this case, we can, of course, produce the light without any light excitation, just with a simple application of a voltage on this electrode, typically 1.2, 1.3 volts, and the presence of a sacrificial agent like the tripropylamine. So this is the mechanism that Alan Bard proposed, and this is a mechanism that is used with beads or with a system that work on this ELISA system. So now, just to give you an idea that the labels that are immobilized have no direct electron transfer, there is a limiting factor that, of course, is the tripropylamine oxidation and also the lifetime of the radical that is very short. So typically, we have to use very large excess of tripropylamine in order to promote this cycle. And as I mentioned, this tripropylamine is toxic and volatile. And if you want to know more, there are beautiful examples of reviews that I put in this slide, if you want to take a picture. So at this point, the question is, what can we do in terms of a real device? Because I have to introduce the device. And what Roche uses is a very simple technique that is based on magnetic micrometer beads technology. And together with Hitachi company in Japan, they develop a system for which essentially they have a magnetic beads that are micrometer in size, where they bind the antibody of interest, and then the analyte will be sandwiched, as I showed you before, in between. Why magnetic beads? Because in this way, with a magnet, they can align indeed all the particles, so they will have always the same number of particles on the electrodes. And then they add the tripropylamine, they apply the voltage, they detect the light that will be, of course, proportional to the amount of analyte that is sandwiched in the antibody. So the question was, what type of system can we maybe improve in terms of design? Well, like I said, ruthenium 3BP is, I would say, the standard that was modified then with sulfonate groups in order to increase the solubility in water. And not only that, the fact that it's negatively charged versus cationic also reduces the background because it reduces what we call non-specific binding to the proteins. So it's important that these compounds becomes negatively charged. And together with Roche, we decided to go from ruthenium to iridium in order indeed to have a better system that could eventually emit more. But at this point, we were wondering, well, can we increase sensitivity just increasing the number of labels? Because this would be very easy. I mean, instead of attaching a single ruthenium label to an antibody, since the antibody is a large molecule, I can attach many ruthenium, or I can have a polymer with many ruthenium. Indeed, this has been done. But when we have done this, and we call this polyru, and this is indeed the standard for Roche, we end up to a big problem. And the big problem is that in the moment we have many ruthenium close by on a single antibody, this excited ruthenium is quenched by its neighbor ruthenium because it's either oxidized or reduced. And you know very well that electron transfer, when we have a very close distance, will be very efficient. So in reality, what happens is that instead of having 6 times the signal that you would expect, we had only 3 times the signal we would expect because of this very efficient quenching of the neighbor ruthenium. So then we thought, okay, then we will make a very small particle, just few nanometers. We use silicon nanoparticle. And with this, we could make sure that the curvature of the particle will make the ruthenium quite far apart, so that the quenching will be less prominent. So we did this, again, with Roche Diagnostics, we patented, but again, the number of units were not, I would say, proportional to the signal we would expect. On top of that, you have to imagine that now I have to functionalize the particle with a single attaching point in order to attach to the antibody. So at this point came an idea, and the idea was, well, what about if we make free the emitters in order to avoid the quenching? So in other words, can I imagine that instead of having everything covalently linked, I now join all the system, the emitters, through a dynamic covalent bond, like the disulfide bond. And disulfide is particularly interesting because in the moment we reduce the ruthenium, we will also reduce the disulfide to thiols. So what we will do essentially, we will break the system and make all the ruthenium or iridium free in solution, avoiding the quenching. So we will have an amplification that is finally proportional to the number of labels. And this is indeed the case, and of course you can imagine that this can be done directly to the magnetic beads or to any other beads in order to introduce hundreds of these ruthenium units that contains indeed our disulfide bond. So will this work on a real system? And of course the answer is yes, and I will show you now 2 examples. And in particular, I will show you the sample indeed on the beads, exactly the same beads that Roche use, that are these magnetic beads, where indeed we can make free all the chromophores, so we can really amplify the signal hundreds of times when we bring our magnetic nanoparticle on an electrode And then, by an electrical stimulus, we will break the disulfide system, since we will reduce the disulfide to thiols. So this was the idea. How can we monitor this process? And here I have to really thank Giovanni Valenti, with whom we work on this project, because he devised essentially a microscope where we could see every single beads using a microscope by applying indeed this electrical signal. So it's a very, very simple device where essentially you will see in a moment we have the electrode here on the bottom, and then by the microscope we can really visualize the emission of the single beads. So what do we see at this point? What we see is what perhaps you already are thinking about, because in the moment we have the covalent bonding of the ruthenium to the beads, like in the case of the Roche system, we see all the beads, of course, lighting up. And we will have a certain signal in ECL. But in the moment we have the ruthenium bound to the disulfide bond, as soon as I apply the voltage, I will reduce the disulfide to thiols, and I will make the ruthenium to diffuse into solution. And so what you will see that already in milliseconds, you have this increase of the signal due to the diffusion of the ruthenium on the surface of the electrodes. So what is happening in terms of mechanism? So let me spend 2 minutes on the mechanism, because it's very important. We have, for the first time, a really switch in the mechanism. So what I told you at the beginning, and what we was studied for many years in regular immunoassay, was that when we have a small molecule on the electrode and we apply a positive bias, of course you know that the ruthenium can be oxidized from ruthenium 2 to ruthenium 3. At this point, if it is really in contact with the electrode, we have what we call catalytic cycle. So the tripropylamine that can be deprotonated can interact directly with this ruthenium 3+ to form the excited state. This is the best mechanism one could imagine. But now with the beads, we are not in contact with the electrode. We cannot do that. So with the beads, what we will have is indeed a different story, because as I mentioned to you, we cannot have any direct electron transfer. And so this catalytic cycle cannot work. So it's the tripropylamine that will reduce the ruthenium 2 to ruthenium 1, and then eventually to reoxidize to the excited state. And here we have a lot of problems, and this is why we need so much tripropylamine, and the efficiency is not so high. But what is happening in our case? What is happening is something very beautiful, because in the moment I have, of course, our system covalently linked, I am in this second case, so I need a lot of tripropylamine in order to turn on the emission of the ruthenium. That is what Bard described for the antibody system. So essentially I have a complicated system where the efficiency is quite low, because I have to go through this entire process. While if I have a system that I can break at the same voltage, now the ruthenium will be in contact with the electrode. So I go to the self-catalytic system. So I can essentially light up all the ruthenium that are essentially made free during the process. So this is quite exciting, but let me show you now the real example with a real system. And the first things we measured was indeed what happens if we now can reduce the tripropylamine and have much less, because if it is a catalytic system, we don't need so much tripropylamine, we don't need to circumvent this problem. And indeed, we see that we have an increase of 600% in the signal already when the tripropylamine concentration is only 50 millimolar. This is a big advantage, not only because we use less material, but because, like I said, it's a very toxic and volatile system. But the second thing I want to show you is indeed on a real assay. So what will happen if we reproduce the regular immunoassay that many people in Roche itself is doing? So with an antibody where we attach our system, our luminophore, and so on and so forth. And so in this case, we decided to really look at very low concentration of this analyte. And so as you can see here, at very low concentration, we are talking here about femtomolar concentration, we don't see the beads at all. I mean, we cannot light up anything. But in the moment I have this ruthenium that can be cleaved, and I have this particle that can release hundreds of ruthenium, you see that I can really detect the signal. But not only that, let me show you an example with antibodies that is the most interesting things. So in this case, we again have the magnetic beads with streptavidin, and then we have the biotin on the antibody of interest that, of course, is connected to the ruthenium luminophore. And you know very well, streptavidin and biotin forms a supramolecular bond, a combination of systems. And now we should be able to really detect these particles because, of course, the ruthenium that is attached to the antibody will be connected to the magnetic beads. To do that, I have to tell you that we modify the design of our chromophore because we compare again the fully covalent system with a disulfide bond, but also we had to increase the length in order to make sure that this coupling with the antibody was efficient. So what do we measure in this case in a real example with a real antibody? What we measure is something beautiful because you see that we can go down 10 times in the concentration of antibody that we can detect. So at this point, you wonder, okay, so can we go further? I mean, can we imagine now, instead of having just a simple analyte or to do immunoassay or antibody, can we use the same system to detect a pathogen? So to capture a virus or a bacterium or an infectious, any type of infective system. And of course, this is the idea of the company that just found. And the idea is that we just collect the sample. I will show you that we collect just mucus. And we don't do any pathogen isolation and enrichment. So essentially, we go straight from the sample directly to the electrodes using this technology. And through the fact that we can have, for each pathogen, hundreds of these labels, we can amplify the system and having essentially a kind of selectivity and I would say detection limit that is as much or even higher than PCR, but in minutes, because essentially we are not essentially extracting any DNA or RNA from our system. So how does it work? Exactly like I described. So you can imagine that now our magnetic particle is, of course, equipped with an antibody that is able to recognize the spike proteins on the surface of the SARS-CoV-2 virus. And then, of course, we use the same antibody that is attached to the ruthenium labels. So very, very simple system. Just you mix together, and then you look at the emission of the particle. So what do we see compared with the regular, say, ruthenium compound? So that when we do not have the disulfide compound, so we have essentially only the covalent system, we still get, of course, a very nice signal coming out from this sandwich system. But as soon as we have the SS bond, so we can break the bond and make free the ruthenium, you see that the detection limit is much, much lower, and we can go from 1,700 to 1,100 virus detection per milliliter. So here we are essentially at PCR, but on something that we don't require any treatment of the sample and can be done in minutes. So we are very, very happy about this, of course. So if you are interested, this was just published, so you can read more the details and how we couple to the magnetic beads and how we do the experiments. So with this, I would like to conclude, because I promised to leave at least 5 minutes for questions. And I think I show you, well, different things, but they are strongly connected by the idea that all the systems we make are responsive. They can either be seen with light, or the process can be monitored by light, or we can indeed produce light to detect something like a virus or a bacteria. So I want to thank the people. In particular, I would like to thank the people that you see here in circle that are working on the project, for instance, for the bacteria detection, like Luigi and Lorenzo, together with Giovanni, and Nicolò and Pablo are also working on this project. I would like to thank Tania for the work on the platinum on the Hydra, together with Claudia Tortiglione. Oops, sorry, I was too fast. And I would like to thank all the group, and of course Giovanni Valenti for the work on the applied part on the COVID detection and on the electroluminescent devices. So with this, I would like to thank also, of course, the money, and in particular 2 projects that allow us to spin off, indeed, the company. So before was a FET Open, and now it's a Pathfinder that will end in December. And of course, I would like to thank you for your kind attention.
Thank you, Luisa, for the very interesting talk. We have time for some questions. Does anybody have their hand up? I can see.
Yeah, there is
yes.
one there. It's hard to see with the light.
Luisa, beautiful results. Congratulations.
Thank you.
Now, this ruthenium tris-bipyridine that you use for the detection of the pathogens, is there any chance that you could use any metal-free organic emitter?
Yeah.
Because there's a similar direction, of course, in OLEDs currently, and that is what people often ask then. Or is that really selectively useful in that regard because also of the electrochemical window that you need there?
Yeah, so indeed we have a project on metal-free ECL. So in this case, we use charge transfer system. So essentially, we use the idea that in the same molecule, if you have a donor and an acceptor, you can generate by charge recombination the excited state. The biggest difference is that the ruthenium is very robust in terms of stability when you oxidize and reduce. So it allows us essentially even to do more cycle. Normally we do only one cycle, but in principle you could cycle several times. So this is why everybody's using ruthenium or iridium. It's not a case, I would say. But yes, so there is no reason why we should not be able to work purely organic with the ECL and then change color. Because of course the ruthenium is always red no matter what we do. The iridium is more flexible because as we show, we can have green, red, blue and so on. So for doing multiplexing is more interesting. Okay. So I agree with you that the organic would be also a possibility.
Okay, nice. Thank you.
Yes. Ben. Oh, there was...
Yeah, wonderful results. Congratulations,
Thank
Luisa.
you, Ben.
Wonderful. You mentioned also iridium instead of ruthenium.
Yes.
It was not clear to me. Is iridium not very useful? Or what is the reason
No, it is useful.
that you continue with ruthenium?
No. We are working with iridium as well. The problem is synthetically is much more challenging. Oh, okay. Because iridium is, as you well know, likes coordination that leads to neutral compounds, and of course neutral compounds are used normally not soluble in water. So we have to introduce these sulfonate groups, and that's what we have done and patent on the structure. And this is synthetically very challenging, so it takes several steps to introduce the sulfonate groups in order to make it negatively charged and also useful for ECL. The beauty is that we can have any color. So we can have a blue, green, yellow, orange, and so on and so forth. Of course, we have to change then the co-reactant, because this co-reactant that I show is designed for those specific windows of the oxidation and the reduction properties. So we have then to go to other type of co-reactant. So we are also studying indeed the different type of co-reactant at the moment. So, but iridium is very, very useful. So we use it, and we have many iridium water-soluble compound.
Can I ask another small question? Please. We also work with organisms, with our photopharmacology approach. And related to that, I often get a question, where do your molecules end up?
Yeah.
So when your ruthenium is liberated, you get this beautiful amplification,
Yes.
But I can imagine this ruthenium complexes also end up binding to other targets in cells and whatever.
Yeah.
So is there a danger that you miss something, or that you have some mismatch of signals and so on?
Yeah, so in the case of the Hydra, we work with the platinum because of the self-assembly, but in principle, yes, you could work also with this type of compounds. And there is a group indeed in France that is Neso Socic, who is doing ECL in tissue and in cells. Yeah. So we are collaborating indeed, is one of the partners of our project. And so in principle, you could use the same thing. So, and you could imagine that in the moment you cleave indeed the ligand, you could even drive the ruthenium in a different cell part.
Yeah.
So this is something we are studying, not electrochemically, but at the moment we are studying it just by essentially internalization of this different compound with different groups. And I can tell you, that, for instance, the fluorinated compound of the platinum goes into the nucleus, but you phosphonate the pyridine, and then it goes to the mitochondria. So it's very interesting. So you can essentially have a selectivity for the different organelles.
You can tune where it ends up.
Yes.
Oh, that's great. Thank you so much.
Thank you.
Last questions?
Thank you. Thank you, Luisa, for this very nice recent results. I was just wondering, in your experiments with the small animals, it seems that the introduction of the platinum is accelerating their growth. Did you check for the possibility to do the same experiments in the dark, fully in the dark?
Yeah. So in this case, indeed, we are not really irradiating the animals. So essentially, this effect of the regeneration is in the dark. So we use the light just to see where the platinum goes. But it's not triggered by the light. It's triggered by the self-assembly. And this is proven, we just submit the paper, because if we have compounds like with adamantyl units, so where the self-assembly cannot occur, so the platinum will be as monomeric species in solution, we don't have any, we could see the compound inside, but we don't have any biological effect. So it's really the fact that when the platinum starts to self-assembly and we measure the electrochemistry of the self-assembled system, the platinum oxidation shifts almost 1 volt. So essentially, it's almost platinum 3. So it's able to go from platinum 2 to platinum 3. So essentially, it's able, in the moment self-assembly, to donate an electrons. And that's why we think that this strong reducing power of the platinum is triggering all these other enzymatic and then eventually the production of stem cell. So it's really the self-assembly that triggers this phenomena. It's not the light. Yeah.
If there are no further questions, please join me in thanking Luisa for the very super talk.